and then we're going to go to screen sharing, and today we're going to start thinking about cells. So cell notes are posted. Like usual, they're in Word and PDF form if you have a preference which one you want to look at. So in this section, we're going to talk about cell structure and function, and we're going to look at cells in general, but then we're also going to look at how cells communicate with each other. Because if we're talking about organisms like people, where we have a lot of cells in our body, the cells have to communicate with each other and coordinate their activities. And we'll also look in detail at one particular part of the cell, the plasma membrane, and see how things can move through the membrane, either to get into the cell or out of the cell, depending on which side they start on. So we'll look at cells, we'll look at how cells communicate with each other, how things get in and out of cells. And a lot of times communication requires something to get into a cell. And the other thing we'll talk about a little bit in this section is we'll talk about viruses. Remember what we said in the first chapter, viruses aren't made of cells, so they're not considered to be alive. But even though viruses aren't made of cells, for a virus to reproduce, it has to be inside a living cell. So since it has to be inside a living cell, this means that we typically think about viruses when we think about biology. Even though technically viruses aren't alive. To be alive, you have to be made of cells, and viruses are not made of cells. So let's start thinking about cells, and keep in mind something that we've said before. We really want to organize our thoughts about cells around the idea that cells are little organic factories, and what do? They make proteins, and then they can use these proteins to do something. But if we think of cells as being organic factories to make proteins, this will help us organize all the information that we cover. Because everything that goes on in a cell is really organized around the idea of making proteins, using those proteins to do something, and ultimately using those proteins to reproduce. Because remember, reproducing is the ultimate goal of all living organisms. Well, the first person to see cells was an Englishman named Robert Hooke, and he saw cells about 350 years ago in 1653. And he was actually looking at cells in a layer that's called cork, and cork is found in tree bark. So if you've ever seen a cork stopper to a wine bottle, or if you've ever gone fishing and used a cork bobber, it's the same type of cork that's found in those items. Now, what Hooke didn't know at the time was cork cells in tree bark are actually dead cells. All the material inside the cell has basically deteriorated. The only thing that's left is the very outer covering of the cell, something called the cell wall. Hooke looked at these through today what we would call a magnifying glass. Keep in mind they didn't really have microscopes like modern microscopes 350 years ago. And Hooke decided to call these things he was looking at cells. Why? Because he was basically looking at little rectangles or pentagons or hexagons, things with 5 sides or 6 sides instead of 4, but there was nothing inside them. And the reason that Hooke named the cells is because at the time, monks in a monastery lived in rooms called cells, and monks were supposed to take a vow of poverty. So Hooke imagined that if you could climb up on the roof of a monastery and you could see through the roof, you'd see a room with 4 bare walls and nothing else because of this vow of poverty. And since he could see the walls of these things he was looking at, but he couldn't see anything inside, he named them cells. Well, as I say, it turns out he was looking at dead cells, and most cells actually do have a lot of stuff inside, but the name stuck. So these are actually some black and white photographs of drawings that were made by Hooke. Now, one of the problems at the time, as I mentioned, was if you wanted to look at something in the microscope, you couldn't do things like you could do today. Today, if you really wanted to see something through a microscope, you could order a microscope online, or you could go to a store and buy one in a brick and mortar place. Back then, if you wanted to look at really small objects, you couldn't buy microscopes. Nobody made them and sold them. You had to make your own microscope. Now, keep in mind, most of the microscopes of the day were really just what we would call magnifying glasses. So they were a little bit simpler than today. Most microscopes today have 2 or sometimes 3 lenses. And they are magnifying glasses if we look at each lens separately. But if we have one magnifying lens that magnifies things 5 times, and then we pass that same image through another magnifying glass that magnifies things 10 times, we get a total magnification of 5 times 10, of 50. But at the time, they were only using single magnifying glasses. So basically, magnifying glasses like the ones you might have used when you were a little kid, except they were much, much, much smaller. Think of a magnifying glass the size of maybe a contact lens, and you're in the right size range. Well, after Hooke made his observations, this inspired other people to start looking at materials. And people continued to look at living organisms or parts of living organisms through cells for the next 2 centuries. One of the most famous was a Dutchman named Anton van Leeuwenhoek. And he was able to see more through his magnifying glasses than other people were, not necessarily because he was a better biologist, because he was better at grinding glass, because you had to make your own magnifying glasses. So to be able to see things, you had to be able to grind the glass very precisely so that it would magnify what you were looking at without distorting it. And he was able to see blood cells, microscopic algae, bacteria. Some of the other people in his lab saw sperm cells. So he saw all sorts of different types of cells. But there was a problem. When you start to magnify things too highly, you start to see diffraction of what we call white light. And white light is just what we refer to as kind of the color of sunlight, or some room lights are. And when white light starts to diffract, if you had a physics class, you might remember that light is actually made up of the visible spectrum. Sometimes we abbreviate it as RGBIV, red, orange, yellow, green, blue, indigo, violet. When you start to magnify things too much, light starts to separate. It diffracts into all these colors. And when that happens, you can't see anything through your magnifying glass. Today, we actually use a type of microscope called an achromatic microscope. A at the beginning of a word, like in front of chromatic, means not or without. And chroma actually means color. So achromatic microscopes don't diffract white light. So whereas Leeuwenhoek was able to use his magnifying glasses and magnify things so they looked about 200 times bigger than they really were, today we can use achromatic microscopes, and we can magnify things so they look about 2,000 times bigger than they really are. At about 2,000 magnification, though, we start to run into some other problems because of the properties of light. So if we want to magnify things much more than 2,000 times, we have to start using electron microscopes or something like that. And electron microscopes don't use visible light. They basically shoot beams of electrons at cells. And electrons allow us to see much smaller things. Instead of magnifying things 2,000 times, we can magnify things 200,000 times if we're using an electron microscope. So Hooke did his work in 1653. Leeuwenhoek worked towards the end of the 1600s and the early 1700s. But people continued looking at living organisms really for another 100 years or so. And finally, in 1838, a German botanist named Matthias Schleiden proposed that all plant tissues were made of cells. Now, keep in mind, this wasn't really a revolutionary idea because people had been looking at plants or parts of plants for about 200 years at this point, and everything they had looked at had these little cells in it. The cells weren't always exactly the same. They didn't all look like Hooke's original cork cells, but you know, this was something that botanists could easily agree with. And this was the first part of what came to be called cell theory. So the very next year, in 1839, another German, keep in mind that a lot of the work at this time was done by Germans. Why? Because you still can't really buy a microscope. So the people who can see the most through microscopes are people who can grind glass very well. And the Germans are the best technicians in terms of grinding glass at this time. So the next year, 1839, Theodor Schwann, who's a zoologist, well, zoologists study animals. Schleiden was a botanist. Botanists study plants. And Schwann published a monograph, which is basically just an article or a paper. And he focused on what were the differences between plant and animal cells. But he also said that all cells were basically the same and all living things are made of cells. Now, keep in mind, at the time, Schwann studied animals. He's a zoologist. Schleiden studied plants. He's a botanist. Today, we know that we don't put bacteria in the same group of living organisms as plants or animals. We don't put fungi in the same group as plants or animals. But if you were alive back in 1838, all living things were considered to be either a plant or an animal. So when they said that all plants were made of cells, and then the next year all animals were made of cells, that meant every living thing was made of cells. Now, the second part of cell theory wasn't added till 1855 by another German, Rudolf Virchow. And Virchow said that all cells come from other cells. In other words, they don't just pop into existence magically. And this might seem like an obvious idea to us, but keep in mind, at the time, a lot of people believed that cells could just pop into existence out of nothingness. If you go back and you read the diaries of educated people in the 1600s and 1700s, and even into the 1800s, there are recipes for making living things. One recipe that I've read is if you want to make mice, if you want to make living mice out of non-living material, here's how you do it. You take a pile of dirty laundry and throw it into a corner in your basement and put in some bread crust and some cheese. And if you come back in a week or so, there will be mice in your laundry. So you've made living mice from laundry and bread crust and cheese. Well, I mean, today we would say that, you know, a mouse climbed in through an open window in your basement and found a nice warm place with some food, but you didn't make mice out of laundry. The mice crawled in. But by the 1800s, people were getting a little smarter. And a lot of people said, well, it's pretty obvious. You can't make things like mice out of laundry. But on the other hand, people were starting to look at things through microscopes regularly. And with a microscope, it's very similar to using the camera on your phone. If you go to take a picture of something with your camera and it's out of focus, you don't see anything. You just kind of see a blur. But then when it comes into focus, you can see whatever you're looking at sharply. And the same thing is true with a microscope. So what people were discovering when they looked through a microscope is, if you look at a little drop of pond water, it doesn't seem to have any living organisms in it. Now, that's when it's kind of out of focus and blurry. But then when you play with it, when you get the pond water into a sharp focus so you can see it clearly, now you can see little organisms swimming around in it. So a lot of people thought, well, maybe you can't make big living organisms like mice, but maybe you could make small cells. They're microscopic. Maybe they could just pop into existence. So Virchow said, no, every cell comes from another cell. It doesn't just pop into existence. So these are the two basic ideas of cell theory. All living organisms are made of cells. And remember, that was one of the things that we said was a defining characteristic of living organisms back in the first chapter. And the second part is cells come from other cells. They don't just pop into existence. Today, we kind of express the same thing in a different way. We say that cells are the unit of structure and the unit of function of living organisms. So this basically says living organisms are built from cells. When we talk about structure in biology, we're talking about the parts of something. And if we want to understand how living organisms function, we have to see how the cells in those organisms function. Now, there are two main types of cells. There are millions of living organisms, different species, but there's really only two major groups of cells. One is what we call prokaryotic cells. And prokaryotic cells don't have a nucleus. We'll talk about what a nucleus is in just a few minutes. And they don't have membrane-bound organelles. And we'll also talk about those in a few minutes. Now, there's two major groups of living organisms that have prokaryotic cells. One is a group that we call the bacteria, and one is a group that we call the archaea. Archaea means ancient, like archaeology is the study of ancient cultures. And actually, the archaea used to be classified as a subgroup of bacteria. And so it kind of makes sense. Their cells are similar. However, in recent years, when our techniques have improved and we can study not just cells, but we can study chemistry, it turns out that some of the metabolism that goes on in archaea is very different than some of the metabolism in bacteria. So for that reason, archaea are put in their own separate group now. The other type of cell is eukaryotic cells. And eukaryotic cells do have a nucleus. Now remember, we talked about the nucleus in atoms a few days ago. So this is the same word, but it has a different meaning. And whenever we talk about the nucleus of something, it's always kind of the central part, the core. In the case of the nucleus in our cells, we'll talk about it in more detail later, but the most important thing to remember about the nucleus is this is where the chromosomes are. This is where the instructions for making DNA is. Now, let's think about membrane-bound organelles for a moment. If we look at a typical cell, one of the things we see is that every cell is surrounded by something called a plasma membrane. It's sometimes called a cell membrane, but we'll refer to it as a plasma membrane. And this plasma membrane is really two layers of lipids. We'll look at the structure of the plasma membrane a little bit later. But eukaryotic cells also have membrane-bound organelles. So what's a membrane-bound organelle? Well, an organelle, E-L-L-E, means a little something. So this is a little organ. So it's kind of like, think about the organs in your body, the stomach, the brain, muscles, things like that. So this is a special part of a cell that does some task, like your stomach helps to digest your food. When we look at these organelles, they're separated from the rest of the cell by a membrane. They're surrounded by a membrane. So these include things like the smooth and rough endoplasmic reticulum, the Golgi apparatus, some things called vesicles, lysosomes, peroxisomes, chloroplasts, and chloroplasts are really one type of something called a plastid. Mitochondria and then all sorts of vacuoles. So these are the main membrane-bound organelles. And one of the reasons that eukaryotic cells are divided into all these little compartments that are separated by a membrane is when we look at prokaryotic cells, prokaryotic cells are much smaller. A really big prokaryotic cell is only about the size of a chloroplast or mitochondrion. So we'll come back here in a minute, but let's skip down to look at a eukaryotic cell for a second to get an idea what we mean. So here's a diagram of a eukaryotic cell. This one happens to be an animal cell. And this little kind of purplish thing that's got the squiggly lines in it is something called a mitochondrion. This one is sliced in half so you can see the inside. Here's one over here that's only sliced in half for the front half. So a really big bacteria cell is about this size, whereas a eukaryotic cell, that's this whole thing. So notice that eukaryotic cells are much larger than prokaryotic cells. And there are some pluses and minuses to this that we'll talk about. So when we look at cells, what we want to keep in mind is prokaryotic cells are much smaller. So that this is going to mean that most things that happen in prokaryotic cells happen in a relatively simple, basic manner. There's usually a lot of similarities without the same processes occur in eukaryotic cells, but things tend to be more complicated in a eukaryotic cell. So let's look at a prokaryotic cell. So here we have a diagram of a prokaryotic cell. And let's think about what the basic parts are, what they do. And if you're following along and taking notes, the description of what the parts do is down a little bit lower in the notes. Towards the central part of each prokaryotic cell, we have an area called the nucleoid. Oid, when you see it used, means like something else or similar to something else. So this is similar to the nucleus. It's not a nucleus. We'll see later that the nucleus has a different structure, but it's similar to a nucleus. Now the reason it's similar to the nucleus is this is where most of the DNA in the cell is. And the DNA is organized into chromosomes. Now, prokaryotic cells only have one chromosome. So the chromosome is this kind of twisted up purplish thing, and the area of the cell where it's found is the nucleoid. Now, having only one chromosome is unusual. Does anybody know how many chromosomes a person has? How many do you have? Most likely, anyway.
26.
12.
23, somebody said, and you have 23, but 23 pairs. Because, in fact, your chromosomes are numbered, and you get one version of chromosome number 1 from your mother in her egg cell. You get one version of chromosome number 1 from your father in his sperm cell. And when we look at eukaryotic cells, they almost always have an even number of chromosomes. They don't all have 46 like people do. Some types of living organisms, their cells have more chromosomes. Some living organisms, their cells have less. But it's almost always an even number. And this is because of something that we'll talk about later in the semester. Eukaryotic cells can reproduce sexually. They can have two parents, the mother and father. Prokaryotic cells only have one chromosome. So one of the things this tells us is they don't reproduce sexually. The way that bacteria cells, that prokaryotic cells reproduce, is they eat some nutrients. They get bigger until they're about twice their original size. They make a duplicate of their chromosome, and then they split in half. And each half gets one version of the chromosome, but they don't reproduce sexually. If we look at eukaryotic organisms, there are hardly any that have an odd number of chromosomes. Because if you get one version of each chromosome from your parents, each parent, 2 times anything is an even number. Now, if we look at this chromosome in the prokaryotic cell, it's made of DNA. So the fact that it's made of DNA and only DNA is also another difference that we'll see when we start looking at eukaryotic chromosomes. So eukaryotic cells usually have an even number of chromosomes. Prokaryotes just have 1. Eukaryotic chromosomes are going to have some material besides DNA, but prokaryotic chromosomes only contain DNA. And then it's hard to tell from this picture, but remember when we talked about carbon, we said that carbon could form 3 basic shapes. It could form straight chains, it could form branch chains, and it could form rings. Well, this big molecule of DNA in here is actually a ring molecule. Now remember, DNA is made of smaller units called nucleotides. In the chromosome in a prokaryotic cell, the first nucleotide is bonded to the second nucleotide. The second nucleotide is bonded to the third. And when we get to the last nucleotide, it's bonded back to the first one. So we have a kind of a ring structure. Now, if we look at what the chromosome does in prokaryotic cells compared to what it does in eukaryotic cells, they both do the same thing. If we look at the DNA in this chromosome, the DNA can be divided up into little sections. And the sections are called genes. And each gene is the instructions to make a protein. Now, a typical bacteria cell makes about 10,000 proteins. So that means there's about 10,000 genes in here. The smallest bacteria that's been discovered, or I should say the one with the least DNA, has only about 4,200 genes. And there's actually one lab that's trying to use, trying to do a process called knockout genes. In knockout genes, you can deactivate a gene. And what they're trying to do is they're trying to take this bacteria with 4,200 genes, and one at a time, they're trying to knock out each gene and see if the bacteria can still survive. What this lab is actually working on is kind of what's the simplest organism, what's the least DNA you could have and still have a living cell. You know, the smallest organism that exists in nature is this one with 4,200 genes. But could we get by without a few of those? Nobody knows until we ultimately get some results. So our DNA is the instructions for making genes. And remember when we, for making proteins. And remember when we say it's the instructions for making proteins, what we're talking about is it tells the cell the primary structure of the protein. And the primary structure, if you recall, is the order of the amino acids. So this is why I think I told you before, sometimes the DNA is compared to a spelling book. Spelling book tells you how to spell a word. The DNA tells the cell how to spell a protein, if you think of the amino acids like letters. Now, the DNA has the instructions for making proteins, but the DNA doesn't actually make proteins. Remember, we said that RNA makes proteins. And there are actually 3 main types of RNA involved in making proteins. One of the types of RNA is basically made off of the DNA. And this is a type of RNA that's called mRNA. It's a lowercase m. And mRNA, we say, is transcribed from the DNA. And basically what it means is that mRNA contains a copy of the instructions for making the protein from the DNA. So the mRNA starts to be produced, and in a bacteria cell, these little blue dots represent the ribosomes. So as soon as the mRNA starts to be produced, we have one of these little blue dots. We have a ribosome that comes in and attaches to the mRNA. And what the ribosome does is the ribosome has the ability to read this messenger RNA and actually collect amino acids and put them into the correct order. Notice that the ribosome actually moves in right next to the DNA and starts on the messenger RNA, sometimes before the messenger RNA is even finished. It hasn't been completed. And the ribosome starts making protein from the very first part. So let's go down a little bit. We're going to come back to this drawing in a moment, but let's come down and okay. It's too far down. We'll stay here. If we look at our ribosome is made of two little pieces. And one is bigger than the other. So there's one of the pieces that's called the large subunit, and there's one of the pieces that's called the small subunit. And they're both made from protein and RNA. Now remember, we just said messenger RNA is transcribed from the DNA. The type of RNA that the cell uses to build its ribosomes is called rRNA, and the R stands for ribosomal. And that's a lowercase r for the first R in rRNA. So the ribosome attaches to the messenger RNA, and the ribosome starts putting the amino acids in the correct order to build the protein. The problem is, where can the ribosome get amino acids? Well, the ribosome gets amino acids from this material that's out around the chromosome and the ribosomes. And this material is called cytoplasm. And the cytoplasm is kind of a semi-liquid, semi-solid material. So it's kind of got like the consistency of a milkshake. It's a little bit thicker in some places than other places. It's a little bit thicker in some cells than in other cells. But it's not quite solid. It's not quite liquid. It's kind of in between. It is fairly liquid because remember, the most common molecule in the cytoplasm is what? Water. But there are lots of things dissolved in this cytoplasm. There's ATP molecules for when the cell needs energy. There's ADP and phosphates that are the products from when we broke ATP down to get energy. There's glucose molecules to use to rebuild the ATP. There's reactants for all the metabolic reactions that happen in the cell. There's the products of the metabolic reactions after they've occurred. So the cytoplasm is kind of like this big warehouse of all these chemicals that the cell is going to need. So one of the types of chemicals that's in the cytoplasm is loose amino acids. When I say loose, they're not connected, they're not part of another protein. And so when we look at these amino acids that are loose, another RNA picks up the amino acids and brings them to the ribosome and puts them in the correct order. Well, remember, we said the instructions were copied from the DNA onto mRNA. There's rRNA in the ribosome, and the RNA that picks up amino acids in the cytoplasm and transfers them from the cytoplasm to the new protein that's being built on the ribosome. The RNA that transfers them from the cytoplasm to the new protein is called transfer RNA, or it's usually abbreviated as tRNA. And again, it's a lowercase t, just like a lowercase m and a lowercase r. So the RNA is capitalized, but the letter in front of that is always small. Now, there are actually several other types of RNA, but these are the main three that are involved in building proteins in a cell. So in a prokaryotic cell, it's a pretty simple process to build a protein, or we usually say synthesize a protein. We've got some instructions in the DNA. Those are our genes. We transcribe those instructions. We're not going to work with the original copy. We don't want to damage it. This ribosome, which contains some rRNA, attaches to the messenger RNA, and then transfer RNA brings in our amino acids from out in the cytoplasm. Notice there are some enzymes involved because building the protein involves some metabolic reactions. Notice since we're building a larger molecule, this is a type of dehydration synthesis reaction, the type of reaction that we always have when we build a larger molecule. So this is all there is to building a protein. Now, since a prokaryotic cell is so small, it doesn't matter where we make a protein, it's not very far away from where we're going to use it. One of the big differences that we'll notice when we look at eukaryotic cells is they're so big, sometimes proteins are made very distant from where they're going to be used. So it's a little more complicated to get the proteins where we want to get them. Basically, building the protein isn't all that much more tricky. So now let's think of some of the other parts of the cell. Those are the parts that are directly involved in protein synthesis, building proteins. So the plasma membrane is this kind of yellow line, and notice that it surrounds the cytoplasm. And the plasma membrane is made of 2 layers of phospholipids. And we can go down to this isn't too far. So here's 2 layers of phospholipids. Notice that the phospholipids face in opposite directions. Now remember, we built a phospholipid by taking a molecule of glycerol and combining it with a phosphate group and 2 fatty acids. So these kind of orangish round areas represent what are called the polar heads. This is where we have the phosphate group and our carbons with the oxygens. And then remember, fatty acids have a long section that's just carbon to carbon bonds and carbon to hydrogen bonds, which are all nonpolar. So we have 2 nonpolar tails coming out of each phospholipid. Now keep in mind, when we think about the plasma membrane of a cell, one side of the plasma membrane, let's say it's this upper side, faces the cytoplasm. Well, remember what we just said. Cytoplasm is mostly water molecules. So the polar part is against the water, and polar things we know will mix with water. The other side, the lower section of polar heads, faces the external environment, the outer environment of the cell. And cells live in environments, guess what, that have water in them. So the polar part is again on the outside, and we have some water down here. But if we look at most of the thickness of this membrane, most of the thickness is hydrophobic. It has nonpolar bonds. And we know that water doesn't mix very well with things that are hydrophobic, that have nonpolar bonds. So what this means, the water on the inside of the cell in the cytoplasm is trapped. It can't get out. It can't go through the membrane. Why? Because this nonpolar hydrophobic layer is too thick. On the outside of the cell, the water and whatever is dissolved in it can't get through the membrane to get inside, because again, this nonpolar section is too thick. So remember, one of the properties of living organisms that we talked about back in chapter 1 was that living organisms have a very complicated internal chemistry. Well, we don't want stuff from the outside getting in to mess up that internal chemistry, or we don't want stuff from the inside leaking out. So by having this thick nonpolar hydrophobic layer, that keeps the inside of the cell separate from the outside of the cell. So that helps the cell maintain its internal chemistry. So here's our plasma membrane. If we go just outside our plasma membrane to this kind of brightish green color here, this represents the cell wall of the bacteria cell. Now keep in mind, the plasma membrane is mostly made out of lipids. So think of lipids, think of oils. So the plasma membrane is relatively flexible. It has a lot of give to it. It can change its shape. The cell wall is made out of chemicals called peptidoglycans. Peptido, remember we talked about dipeptides and polypeptides and peptide bonds. Peptides refer to amino acids. Peptidoglycan, glycan refers to glycogen, or a process we'll talk about later called glycolysis. So it refers to carbohydrates. So the cell wall is made up mostly of carbohydrates, but the carbohydrates are connected together by amino acids. So the cell wall, unlike the plasma membrane, is actually very thick. It's very stiff. It's rigid. It doesn't have any give to it. And in fact, if we have a bacteria cell that's this shape, then that's the shape of the cell wall. Because one of the things that the cell wall does is it gives the cell its shape. Now, what's the advantage of having this hard, stiff, thick layer, the cell wall? Well, because what the cell wall does is it protects the bacteria cell that's inside. It stops some chemicals from getting through. Some things like detergents or antibiotics that could harm the cell. It stops the cell from absorbing too much water. Some cells absorb so much water that as they absorb water, they swell, kind of like blowing up a balloon. And if they absorb too much water, they can pop, like if you put too much air into a balloon. Well, the cell wall is so thick and stiff and strong. When the cell starts to absorb water and it starts to press out against the cell wall from the inside, the cell wall is so strong it doesn't have any give to it. The cell can't absorb any more water because the cell wall doesn't expand, so no more water comes in. So bacteria cells can't absorb water, at least under normal circumstances, and pop like a balloon. Human cells don't have a cell wall. Human cells actually can do that. Our cells can absorb so much water that our cells pop like a balloon, which of course is not very good for you. Now, outside the cell wall, a lot of bacteria have this lighter green layer, which is labeled the capsule. It's sometimes called a slime capsule. And it's built from polysaccharides, so large carbohydrate molecules, and they actually kind of have a slimy texture to them. And again, like the cell wall, this is for protection. It stops some chemicals from getting into the cell. It can also stop some cells, like cells in our immune system, from getting a good grip on the bacteria cell so that they can destroy it. Now notice we have some stringy things that poke out beyond the cell wall. One of these is a flagellum. Some bacteria cells have a flagellum. Notice they all don't. Some have flagella with an A ending instead of UM. That's just plural for flagellum. So if the bacteria cell had two, they would be flagella. Now, what this flagellum is, it's mostly protein. And it's anchored in the cell wall. And what this does is this spins around like the propeller on a boat motor or a propeller on an old style plane before they had jet engines. So just like a propeller on a plane or a propeller on a boat, bacteria that have a flagellum, or more than one flagella, they twirl this around and they use this like a boat propeller to move. Remember, one of the characteristics that we talked about in chapter 1 for living organisms was that living organisms can respond to their environment. If a bacteria cell can detect chemicals it likes in one direction, can use this flagellum and kind of swim in that direction. If it detects chemicals it doesn't like, it can use its flagellum and swim away from those. Excuse me. The other type of thing that we see sticking out is something called a pilus. Notice when there's more than one, like there is on this cell, pili, P-I-L-I, is the plural of this word. And different cells have different types of pili. But in general, pili are used for attachment. If we have a bacteria cell that's infectious, that attacks other cells, it can use the pili to get a grip on the other cell, to attach to the cell that it infects. But some bacteria cells exchange DNA. Now, when they exchange DNA, they can use the pilus to get a grip on the other bacteria cell so that they don't drift apart, so that they can actually carry out the exchange. And some bacteria cells don't really anchor to another cell, or whether it's a bacteria cell or their host cell that they're infecting. Some bacteria cells use the pilus and they just attach to material. So for instance, some of the bacteria that cause dental caries, which we usually just call cavities, some of the bacteria that cause dental caries can actually attach to the enamel of your teeth and form something called a plaque. Now, they don't necessarily attach to a cell in the tooth. They can just attach to the enamel. And the enamel is kind of like bone, so it's not all living cells. It does have some cells in it. So when we look at a bacteria cell, it could have different types of pili. Some microbiologists give the different types of pili different names, depending on whether they're attachment to another bacteria cell or whether they're for attachment to a human cell that they're infecting, or whether they're attachment for something that isn't cellular, like enamel on a tooth. We're not going to worry about all those different names. If you go on and take microbiology, though, you'll most likely hear about those. But for our purpose, we just want to realize these pili are for attachment. So remember what we keep saying and what we're going to emphasize. Doesn't matter whether we're talking about eukaryotic or prokaryotic cells. What are they? They're organic proteins, organic factories that make proteins. So this next section is really our description of how the prokaryotic cells make proteins that we talked about in class. So here's our plasma membrane. So let's look at this plasma membrane in a little more detail. So here's a diagram of the plasma membrane, similar to the one we have up here. The colors are a little bit different, but we have our polar, or notice this diagram labels them as hydrophilic heads. And we said one of those layers is next to the outside of the cell. The other layer of the hydrophilic heads is next to the cytoplasm. And then this yellow represents those non-polar tails, the hydrophobic portion in the middle. However, if we look at membranes, they don't just have phospholipids in them. They also have proteins. So all these purple blobs represent proteins. Now notice some of the proteins in the plasma membrane, like this one here in the front of this diagram, go all the way through from one side to another. Let's make this a little bigger because this is a tiny drawing. So some of the proteins go all the way through. Some of the proteins are stuck in one layer. Like notice this one is stuck in the outer layer. Some of the proteins actually kind of float on the surface of the outer layer or on the surface of the inner layer. If we look at the proteins in the plasma membrane, notice that the proteins have a similar chemistry to the phospholipids. If we look at the parts of the proteins that are out on the edge, so all these sections kind of sticking out, these are parts of the proteins that have mostly polar or ionic R groups, and they're hydrophilic. If we look at the part of a protein that's in the middle of the membrane, the parts in the middle have mostly nonpolar R groups that make them hydrophilic. So again, the portion in the middle, whether we're talking about a protein or a phospholipid, is hydrophobic. The portion on the outside next to the water is hydrophilic. Now, what do the proteins do? They do all sorts of different things. Some of the proteins that go from one side to another actually have a kind of a hollow core. And so they're like a doorway or a channel. There's usually kind of a flap on the protein, so the channel can be open or closed. But if they're a channel, this means they could let stuff into the cell or let stuff out of the cell. Now, although we could think of a protein that has a channel through it as being like a doorway, or you could think of it like that tunnel that football players sometimes run through to get into the game, there is one big difference. You know, if we look at a doorway, anything can come or go through that doorway pretty much. Might have to be, you know, below a certain size, depending on the size of the doorway. But if this protein goes all the way through, if it's a channel, it's got kind of a hollow inside pathway, only things that have a compatible chemistry can get through. So not everything can get through. So if we look at nerve cells in your brain and your spinal cord and your nerves, they have some channels that sodium can go through. Sodium ions with a positive charge. They have different channels that potassium ions can go through, because the sodium channels don't let potassium through. The potassium channels don't let sodium through. So remember what we keep saying, everything works by its three-dimensional shape, and that's really related to its chemistry. So besides forming a channel, remember one of the functions we already talked about with proteins is they sometimes act as a receptor. Sometimes molecules on the outside of the cell can attach to the protein. Now, when that happens, sometimes the molecule that attaches gets carried inside. Some proteins, like this one here that's basically in the outer layer, can swivel. And after something attaches to them, they can swivel and drop something off on the inside. Some proteins pick something up, and if they pick something up on the outside, it changes their shape. And then when it changes their shape, that bumps against another protein on the inside of the cell. But overall, the best way to think about these proteins in the membrane is the phospholipids basically keep things from getting in and out, because most things can't get through that thick nonpolar layer. The protein's job is mostly to move things in and out of the cell. They do it in slightly different ways that we'll talk about later, but when they move things in and out of the cell, what this does is this can affect the metabolism of the cell. They can eliminate waste products. They can bring in reactants for chemical reactions. So the phospholipids stop stuff from getting through the membrane. The proteins help stuff get through the membrane. But there's different proteins for different stuff. One protein doesn't fit all different materials, kind of like one size doesn't fit all. So proteins are going to move stuff through. Now, when we talk about other cells, we're going to see that there are some other types of molecules in other cells, but those are the main ones in the plasma membrane in bacteria cells. Now notice when we look at bacteria, there are thousands of species, but there's only 3 basic shapes for bacteria cells. Some bacteria cells are round, like the kind of bluish purple ones here. This is called a coccus shape. You might have heard of a type of bacteria called Streptococcus that causes strep throat. Or you might have heard of a bacteria called Staphylococcus that causes staph infections. Or you might have heard of a disease called MRSA. MRSA is methicillin resistant. Methicillin is an antibiotic. MRSA is methicillin resistant Staphylococcus aureus. And does anybody know what MRSA does? MRSA. MRSA is sometimes referred to as the flesh-eating bacteria disease. A lot of people have this type of bacteria on their skin, and it doesn't bother most people. But some people are very sensitive to it, and it can start eating their tissue. I had a student a few years ago, and he was missing the calf muscle on one of his legs. He had had a MRSA infection, and it basically ate his calf. So when we look at bacteria cells, we want to keep in mind that they're simpler than eukaryotic cells, which is the type of cells we have. So sometimes we think that means they're not as good as eukaryotic cells. But keep in mind, bacteria cells can do something that your cells can't. If we take a bacteria cell and we separate it from all the bacteria cells around it, every bacteria cell can survive just fine on their own. They don't need other bacteria to reproduce. They can only reproduce by themselves. They don't need other bacteria cells to survive. If we take cells out of your body, individual cells can't survive on their own. A brain cell or a cell from your heart muscle or a cell from your skin can't do everything that a cell needs to be able to do to survive. It's not a problem in your body because different groups of cells specialize and do different jobs. But if your cells were on their own, they couldn't survive. Bacteria cells, no problem. So in that sense, they are somewhat superior to eukaryotic cells. They can survive on their own. Our cells can't. Now we're going to look a little bit at eukaryotic cells. So remember what we said, eukaryotic cells tend to be much larger. Keep in mind, this is just a general rule. The very smallest eukaryotic cells are actually smaller than the very largest prokaryotic cells. But most eukaryotic cells are bigger than most prokaryotic cells. Now, because they're larger, they tend to have a much more complex internal structure than prokaryotic cells. So let's come down for a minute. Let's look at a diagram. So this is a eukaryotic cell. This is an animal cell, so this kind of bluish purple around the edge. This is the plasma membrane. Notice all the stuff inside the plasma membrane. We've got the nucleus, we've got chromatin, we've got the nucleolus, we've got the mitochondrion, we've got a lysosome, we've got centrioles, we've got a vacuole, we've got smooth endoplasmic reticulum, the Golgi body, rough endoplasmic reticulum, ribosomes, the nuclear pore, the nuclear membrane, all those different parts. Remember, when we looked inside the prokaryotic cell, what did we see? Not all that stuff. All we had in our prokaryotic cell was one chromosome made out of DNA, a bunch of ribosomes, and the cytoplasm. So we've got a lot more parts when we look at a eukaryotic cell. So we want to keep that in mind. Now, eukaryotic cells are found in 4 main groups of living organisms. They're found in plants, they're found in animals, and they're found in fungi. And if we look at plants, all plants are very similar to one another. And if we look at animals, all animals have a lot of similarities. Keep in mind, they're not all similar to people. We're kind of weird animals in some ways. All fungi have a lot of similarities. But if we look at the last category, protists, they don't have a lot of similarities. If we look at protists, protists are a good example of a common problem we find in science. And not just in science, really. As humans, a lot of psychologists have pointed out that one of the things we like to do is divide things into categories. We like to separate them into different groups. You know, we do this with people. We separate people on the base of gender, race, religion, country of origin, all sorts of things. The problem is, if you try to separate living organisms into categories, nature doesn't always cooperate. So living organisms have to have certain characteristics to be considered a plant. They have to have another set of characteristics to be considered animals. And they have to have still a different set of characteristics to be considered fungi. And here's the problem. There are some protists that kind of resemble plants. They carry out photosynthesis, but they don't have all the characteristics of plants. There are some protists that kind of resemble animals, but not enough to group with animals. And there are some of them that kind of resemble fungi, but not enough to really put with the fungi. Now, one good example of this is there's a microscopic protist, and most of the protists are microscopic, not all of them, but there's a microscopic protist that has chloroplasts. Now, we haven't talked about chloroplasts yet, but chloroplasts are the organelles in plant cells that allow plant cells to carry out photosynthesis. And photosynthesis is one of the main things that separates plants from animals. Well, there's a protist that has chloroplasts, so it carries out photosynthesis. So it's a plant, right? Except when you take this protist and you put it in the dark, the chloroplasts deteriorate, and then this protist starts swimming around eating its neighbors. So depending on whether it's in the light or the dark, in the light it kind of resembles a plant, in the dark it more resembles an animal. So which group do you put it in? It doesn't really fit in with either one. And there are some protists that don't really fit into any of the three major categories. The way to think about protists, I always say, is you know, if you've ever cleaned out your basement or your garage or something, you know, you have a pile of like old kids' clothes and old games, and you have a pile of tools, and you have a pile of your old tax records or something. But you always end up with a pile of stuff that doesn't fit into any category. It's kind of the junk. And that's what protists are. Protists are kind of the miscellaneous category. They have eukaryotic cells, so they have cells with a nucleus and membrane-bound organelles, but they don't resemble plants or animals or fungi enough to put them in those categories. So protists are really interesting because they do some things that we don't think of living organisms as doing, but they illustrate the problem that we run into when we try to take nature and divide nature up into nice, neat categories based on our conceptions. There's nothing that says nature has to necessarily match what we would like it to match. So we want to keep that in mind. Now, when we look at eukaryotic cells, let's look at one of the main structures that makes a eukaryotic cell different from a prokaryotic cell. So here's a eukaryotic cell, and this is mostly its nucleus. Out around the edges here with these little red dots, this is some of the area called the rough endoplasmic reticulum. So this is the nucleus. Let's go back down to our big drawing of the cell. So this is the area that's shown in green and purple in here. So remember what we said. The first thing you have to be careful about is we used the word nucleus when we talked about atoms. And nucleus just means the center or the core of something. So this is the core, this is the crucial area of a cell, but it's not the same as the nucleus of an atom. So this whole thing is called the nucleus. So let's look at some of the parts of this nucleus. All this little stringy stuff in here is called chromatin. And chromatin is really just a different name for the chromosomes. Later in the semester, we'll see why we sometimes give it a different name. Chroma actually means color. And chromosomes, chromatin, got its name because if you put a dye or a stain on the cell, usually the chromosomes absorb a lot of that stain. So they're easy to see because they're brightly colored. So when you look under the microscope, you can see them. Now, there are some differences. If we look at eukaryotic chromosomes, they're linear molecules. What does that mean? Well, that means the carbons are basically a straight chain. Remember that when we looked at prokaryotic cells, they had circular chromosomes. Now, another difference is in eukaryotic chromosomes, the DNA isn't the only type of chemical in the chromosome. The DNA is actually wrapped around some things called histone proteins. And it's kind of wrapped around them the way that thread is wrapped around a spool. And remember, prokaryotic chromosomes only had DNA in them. And then the third difference is eukaryotic cells almost always have an even number of chromosomes. And this is because they can reproduce sexually. And remember, we said prokaryotic cells only have one chromosome. So we do still have chromosomes, but the chromosomes are very different than in a prokaryotic cell. Now, somewhere inside the nucleus, we have an area called the nucleolus. Sometimes there's actually more than one nucleolus. And the nucleolus is actually over a specific spot in the DNA. And it's over the part of the DNA that actually has instructions for making rRNA, ribosomal RNA. And the nucleolus is where a cell starts to put its ribosomes together. Doesn't add the proteins yet, but it starts putting the rRNA molecules together to build a ribosome. And this is very important for cells because remember, if a cell is an organic factory that makes proteins, and the ribosome is the thing that actually makes the proteins, that means ribosomes are pretty darn important. So the ribosomes have a special location where they're built. Cells that are making a lot of protein might have more than one nucleolus. Now, inside, around the chromosomes, around the nucleolus, the fluid in the nucleus is called the nucleoplasm. And notice that the nucleoplasm is basically the same thing as the cytoplasm. It's just inside the nucleus. If you ever saw that old Batman TV show where, you know, everything is, they're going to slide down the bat pole to get to the bat cave. Everything was named Bat something. In the nucleus, everything is nuclei something. You know, we give it a unique name. Now, surrounding the nucleus and separating it from the rest of the cell, we have two thin black lines here. And these two thin black lines represent what's called the nuclear envelope. Sometimes also called the nuclear membrane, but usually we call it the nuclear envelope because remember, if we go back to our plasma membrane a minute ago, our plasma membrane has two layers of phospholipids. That's not what we find in the nuclear envelope. In the nuclear envelope, this black line, I'm going to magnify this a little bit more. in the nucleus, this black line that goes all the way around the purple, that's one membrane. So that's 2 layers of phospholipids. And then this kind of bluish line out here, this one has a red dot in it. This is a second membrane. So this is a second set of 2 phospholipid layers. So there's actually 4 layers of phospholipids between the nucleus, and out here would be the main part of the cell, all the other organelles. So that's why we like to call it a nuclear envelope instead of just a nuclear membrane. Because if you hear membrane, you might think 2 layers of phospholipids. And there's not really 2 layers, there's really 4. Now notice this outer layer where they show the red dots, this is really a continuation of this layer that has red dots around it. And this is something called the rough endoplasmic reticulum. So notice this layer of the nuclear envelope is really just a continuation of the rough endoplasmic reticulum. Now, for things to get in and out of the nucleus, it happens a little differently than it does in cells. Remember with cells, we said that most things get in or out of the cell because of the fact that they have a protein that helps them somehow get across the membrane. In the nucleus, there are little holes that go through the 2 layers of membrane. And the holes are actually guarded by proteins. And for things to get in or out of the nucleus, they have to go through a nuclear pore. They have to go through this little kind of white pathway that you can see here. Now, keep in mind, since these nuclear pores are guarded by proteins, remember, proteins work according to their chemistry. So this means not just anything can get in or out of the nucleus. It has to have the right chemistry and be compatible with one of these proteins guarding the pores. If it's not, can't get in or out. It's stuck on whatever side it's on. Now, notice that despite the fact that the nucleus looks a lot different than that nucleoid did in prokaryotic cells, and remember we said the nucleoid got its name because oid means like, it's like the nucleus. Why is it like the nucleus? Because it has a chromosome with genes, and the genes have the instructions for making proteins. So there's a lot of differences in the basic structure of the nucleus and the nucleoid, but these little squiggly lines are chromosomes, and guess what? Just like in a prokaryotic cell, you can divide the chromosomes up into genes, and every gene is the instructions for making proteins. So from that point of view, the nucleus really has the same job. It has the same function inside a eukaryotic cell as it does in a prokaryotic cell. The nucleus, or the nucleoid, is where we have our chromosome, our chromosomes. We might have more than one, but however many we have, this is where we have the instructions for making proteins. So that's the important thing in terms of what they do. Okay, we're gonna quit for here today. So before I leave this section, are there any questions about anything that we need to refer back to some of the drawings or some of the text in here? Okay, if not, I'm gonna stop screen sharing, and we'll see if there are any other questions before we leave for the day. Any other questions? Okay, the chemistry questions that are due next week are posted, so make sure you look at those. If there's something on them you don't understand, you know, send me an email.
I have a question.
Mm-hmm.
We're in chapter 2 and 3?
Yes.
Okay.
Well, actually today we're in the next chapter.
Okay.
So this is chapter 4, right?
Yes.
All right, thank you.
And really, we're going to be talking about different things about cells, probably for the next 4 weeks or 5 weeks.
I do have a question.
Mm-hmm.
I don't really get the part of the genes, the chromosomes, the differences.
Okay, let's go into screen sharing again, and let's go to the whiteboard. Not that my drawings are great. So let's look at a chromosome in your body. So this is the chromosome. And let's magnify this. Now remember what I said, in a eukaryotic cell, there are histone proteins. And the histone proteins are kind of spread out. They're not next to each other normally. So when the DNA comes along, it kind of wraps around this protein clump. Usually it wraps around twice and goes on its way. Now, let's think about taking a little section of this. So keep in mind that if we draw this in more detail, this long stringy strand is the DNA. And then these circles are the proteins. So when we talk about a chromosome, we're talking about this whole thing. When we talk about the DNA, we're not really talking about the proteins that it wraps around. And then every once in a while in the DNA, we can take a little section. This is a section that I've kind of, separated off here. There's a little section that's a gene. And the gene is just the instructions for making a protein. We'll see later that there's some stuff in there that isn't actually used, but this is the easiest way to think it. So a gene is a small section of the DNA. One way to think about it is, a few minutes ago in class, we said a person has 23 pairs of chromosomes. And you have about, not exactly, but you have about 23,000 genes. So that means there's about 1,000 genes on every chromosome. Now, the genes are just small sections of the DNA. One way to think about this is if a lot of times the DNA in a person is compared to a book. So imagine you have a book with 23 chapters. So each chapter is one of your chromosomes. And if this was kind of a do-it-yourself manual, how to build things at home, inside each chapter, there would be 1,000 sets of instructions. So maybe there's an instruction on building a barbecue pit and an instruction on wiring your house so that you can have Christmas lights displays and so you have enough power. But one of the things you have to keep in mind, the reason I didn't draw these genes next to each other is if you look at the human DNA, only about 2% of the human DNA is actually genes. 2% of our DNA is the instructions to make human proteins. The other 98% is other stuff. Some of it is on-off switches that tell the cell when to make the proteins or when not to make the proteins. Some of it, we don't really know what it does, to tell the truth. So think about the difference. The chromosome is the whole thing. The chromosome includes DNA and proteins. But the DNA, if we're just saying the DNA, we're not really including these proteins then. And when we say a gene, we're just talking about a small section of this chromosome. A small section of this DNA. I don't know if that makes it any clearer or not. I'm not sure.
Yes, it does. But yeah, I want to ask a part because the DNA is made up of also the timing.
Adenine, cytosine.
Adenine, cytosine.
Mhm.
How does that explain the part for the like? How does he explain it with the gene also?
Well, if you would look at this, keep in mind. Let's go to a different one. Let's look at our DNA. If we would look at the DNA, what we would see is there's a deoxyribose sugar connected to a phosphate, and that's connected to the next deoxyribose sugar to another phosphate. I'm going to do a couple of these. And so we kind of have a chain of the sugars and the phosphates. Now, the other thing we have is on the sugar, this is where the nucleotide is attached. So maybe the first one is an A, and then maybe we have a G, and then maybe we have a T, and maybe we have a C. I'm just making up that order. The order of these letters, this is what the ribosome reads to figure out what happens, what the order of the amino acids is. So say if you have blue eyes and I have brown eyes, the instructions you have might be AGTC for blue eyes. My instructions might be AGTT, and that might make brown eyes. So really, when we look at our DNA, when we look at a long strand, this is what's really in that strand, if we could magnify it enough, is the sugar and the phosphate and then the bases, and it's really the order of the bases that's crucial in giving us.
I want to ask, through that explanation of that order, is that what we call the gene, since they are
You mean that other 98% I talked about?
No, like the order you said now, because you said it shows what the color of our eyes and stuff.
So that's the part that's a gene.
Okay.
Because really the color of your eyes actually depends on a protein that you make. Kind of like if you have type A blood or type B blood or type O blood, that depends on a protein that you make. So since the instructions for the proteins are in the DNA, that means ultimately, if you have type A blood or type B blood or type O, whatever, it depends on the order of these 4 letters in your DNA.
So that is a gene, right?
Yeah, so that would be a gene, except keep in mind in real life, most instructions for proteins in the human body are a couple of 100 nucleotides long. So there's a couple 100 letters that make up the instructions. So I didn't want to make it too long, you know, for our example, but really the instructions for blue eyes or brown eyes or type A blood or whatever we're talking about would be several hundred, maybe even several thousand nucleotides long. So think of it kind of like a matter of scale. You know, when you look at this, you're kind of looking at houses. Whereas when you look at the whole DNA, instead of looking at individual houses, you're looking at a whole street in some city. And then when you think about all the chromosomes in the nucleus, you're looking at all the streets in some city.
Yeah, thank you.
So it's kind of when you look at a chromosome, all these levels are there. It's just that some of them are not very obvious. Any other questions?
Thank you very much.
You're welcome. Okay, if there are no more questions, I'll let you go then. I'll stop the recording and I'll post that whenever it gets translated into an MP4. So enjoy the rest of your day.